Hello and welcome back. In this lecture we will continue our journey through Chapter 2, the cells. Last lecture we focused on the differences between prokaryotic and eukaryotic cells and journeyed through some of the organelles found within eukaryotic cells. In this lecture we will focus on the internal structure support of cells, also referred to as the cytoskeleton, and talk about cell junctions, which is the way cells can stick together. Alright, let's get right to it. You can see the cytoskeleton highlighted in the picture on the top right. It's all the fibrous looking material that is highlighted green. The cytoskeleton is composed of several different components, which we will talk about shortly. The role of the cytoskeleton is to support and maintain cell shape, hold the organelles in position, move the organelles around. Cytoskeleton is involved in cytoplasmic streaming and interacts with extracellular structures to anchor the cell in place. The three components that make up the cytoskeleton are microfilaments, intermediate filaments, and microtubules. All three of these components have very different functions that we will discuss. Microfilaments help to make a cell or parts of the cell move. Microfilaments determine the cell shape or are made from actin monomers. Microfilaments have what is called dynamic instability. This means they are easy to break down and put back together again. Top picture is a diagram of a microfilament. You can see the little red balls, which are the actin monomers. This structure allows them to be easily broken down. Intermediate filaments are tough, rope-like protein assemblages. These are more permanent and do not have dynamic instability like microfilaments. Intermediate filaments anchor cell structures in place and resist tension and maintain the rigid structure of the cell. As you can see in the top diagram, intermediate filaments are longer than microfilaments, and they are stronger and composed of protein. The last component of the cytoskeleton that we will look at are microtubules. These are the thickest of the cytoskeleton components. They form a rigid internal skeleton for some cells. They act as a framework for motor proteins to move structures within a cell. As you can see in the diagram above, they are thicker than both microfilaments and intermediate filaments. The thickness of microtubules helps give them their strength. First, progress check. This one is asking you to compare the three components of the cytoskeleton. Your book goes into a little more detail than I did, but focus on what we went over here and you'll be fine. Before we get into cell junctions, we are going to focus real quickly on plant cell walls. We as animals obviously do not have this unique structure in our cells. Plant cell walls are semi-rigid structures. They are found outside the cell membrane. Cell walls are made of the polysaccharide cellulose. In this picture here, you can see the thick cell wall of two cells. This important structure has a few roles within plant cells. Plant cell walls provide support for the cell and limit the volume of the cell by remaining rigid. They also act as a barrier to infection and to contribute to form during growth and development. Plasmodesmata, which are unique to plant cells, are membrane-lined channels that connect adjacent plant cells. Plasmodesmata allow for the movement of water, ions, small molecules, and hormones that travel between the plant cells. Cell junctions are specialized structures that protrude from adjacent cells and hold them together. There are three types of cell junctions. There are tight junctions, desmosomes, and gap junctions. An example of a tight junction is on the picture on the top right. Tight junctions prevent substances from moving through the spaces between cells. Desmosomes, as you can see an example in the picture in the middle, hold cells together but allow materials to move between the cells in the space called the matrix. The third cell junction is called a gap junction. These are channels that run between pores in adjacent cells, allowing substances to pass between them. An example of a gap junction is on the top left. The plasmodesmata that we talked about earlier in plant cells is an example of a gap junction. Figure 4.18, pictured here, illustrates all three gap junctions: tight junctions, desmosomes, and gap junctions. Picture 4.18 is a close-up illustrating a tight junction. You can see the proteins that are holding the two cells in place. There's not a lot of room between these cells for dissolved materials to pass through. You can find examples of tight junctions within epithelial cells. Figure 4.18 part 3 here is illustrating desmosomes. These cells are held together, but there is some space for dissolved materials to move between the cells. Last picture here illustrates a close-up of a gap junction. The membrane-lined channels allow the two adjacent cells to communicate with one another and pass materials between them. Alright, here's the last progress check. Take your best shot at these questions and continue on the last few slides. Alright, before we conclude this chapter, I'm going to end on another biology pun. What do you call a picture of a cell? Selfie. Thought this was very appropriate for this particular chapter. Thank you for listening to this relatively short lecture. I wish they could all be this short. This concludes chapter 4. Have a good rest of your day.